The jellyfish Turritopsis dohrnii, nicknamed the immortal jellyfish, is a jellyfish of the order Hydrozoae, living primarily in warm and temperate ocean waters. It is found in the Mediterranean Sea, its original habitat, and in certain areas of the Atlantic Ocean as well. It is also observed in other temperate seas, but it has spread to several regions via currents and maritime transport. Turritopsis dohrnii is a small jellyfish, measuring only a few millimeters in diameter. The species was discovered in the Mediterranean Sea in 1883 and could potentially fight cancer by reactivating G cells and interchanging genes, according to National Geographic's Immortal Jellyfish Swarm, World's Oceans. Besides Turritopsis dohrnii, several other creatures are biologically immortal in the sense that their mortality rate does not increase with age. The hydra, planarian S, the lobster, cone jelly, and certain other jellyfish possess this ability. What does biological immortality really mean? Is it absolute immortality, or an exception to cellular aging? We will explore, step by step, the cellular and genetic mechanisms, and the limits of this apparent immortality. Biological immortality is the ability of an organism not to die from aging. There is no programmed cellular wear leading to death, therefore an absence of senescence. It is distinct from absolute immortality, as the organism remains vulnerable to death by injury, disease, or predation. Absolute immortality is a concept that does not exist in nature. It would imply being both eternal and invulnerable to any form of death. It would be safe from predation, accidents, diseases, natural disasters, and even the end of the universe. It would need neither food, nor water, nor oxygen. A totally invulnerable organism would break the ecosystem's balance. It could not be eaten, and it could not eat. Nature is governed by entropy. Living systems must constantly consume energy to maintain their organization, and this would mean that the organism that could live eternally, without suffering degradation such as wear and damage, would consequently violate these laws. Life is subject to chance. Even if an organism doesn't age, an accident can always kill it. Biological immortality is an evolutionary trick to avoid death by aging, while absolute immortality is a philosophical concept that cannot exist in the material world as we know it. Turritopsis dohrnii is the most famous and most studied species. Its specificity lies in the fact that, once it reaches maturity, the medusa stage, it can return to a juvenile stage, that of the polyp. This unique process is triggered by stress such as hunger, injury, unfavorable environmental conditions, or aging. By going backwards, it escapes aging and can theoretically restart its life cycle indefinitely. It is this capacity, shared with a few other species of the genus Turritopsis, just like Turritops is Nutricolae, that earns it the nickname Immortal Jellyfish. Research is ongoing, and scientists continue to study the genome of these species to understand how this mechanism works and whether it is present throughout the entire genus. P&AS Proceedings of the National Academy of Sciences compared the genome of Turritopsis dohrnii to that of other jellyfish species, including other Turritopses. The results suggest that Turritopsis dohrnii has unique genes, or genetic variations that make its rejuvenation potential particularly high, almost 100% after reproduction, which distinguishes it from other species that might not have the same efficiency or frequency of rejuvenation. The classic life cycle of a jellyfish unfolds in several stages, including egg, larva, polyp, a sedentary fixed form, and finally adult medusa. After reproducing, the adult medusa ages and dies. Turritopsis dohrnii is capable of reversing this process. Instead of dying, it can undergo progress from its mature medusa state to its juvenile polyp stage. This rejuvenation process is not automatic. It is generally triggered by environmental stress or a threat to its survival, such as hunger, injuries, and changes in temperature and salinity in an environment hostile to its survival. In response to this stress, the medusa retracts its tentacles. Its body contracts to form a small mass of cells that attaches to the ocean floor. From this mass, a new polyp identical to the original juvenile version will form, allowing it to restart a new life cycle. First, the standard cycle consists of planula, polyp, then medusa. The planula corresponds to a small swimming and ciliated larva covered with tiny hairs for movement. The planula is born from a fertilized egg and floats in the water. Its main role is to find an appropriate place to attach, generally a solid surface like a rock, shell, or algae. Once attached, the planula metamorphoses into a polyp. The polyp is a sedentary structure that resembles a small coral or sea anemone. It feeds on small particles and grows by forming a colony. This is the phase of growth and asexual reproduction. The polyp can reproduce by budding, creating other polyps, and developing in height when environmental conditions are favorable, including seasonal changes and water temperature changes. The polyp divides through a process called SDR-OB-lot-ion. It segments horizontally, as if stacking saucers. Each saucer detaches and becomes a new entity. Thus, each segment that detaches is an ephira, a small immature medusa. It is tiny, swims freely, and doesn't yet resemble an adult medusa. The ephira grows, feeds, and develops to reach its final form. The ephira transforms into an adult medusa. This is the best known stage of the life cycle, with its bell and tentacles. This is the phase of sexual reproduction. Adult medusae release sperm and eggs which, after fertilization, give birth to a new planula, thus restarting the cycle. Through the process of transdifferentiation, Turritopsis Dohrnii reiterates its life cycle. Transdifferentiation is defined as the process during which a differentiated cell transforms directly into another differentiated cell type, without returning to a pluripotent stem cell state. In the case of Turritopsis Dohrnii, the process combines transdifferentiation and dedifferentiation according to the tissues concerned. We have explained the normal development cycle including fertilized egg, planula swimming ciliated larva, polyp fixed to substrate, budding, ephira young medusa, adult medusa, sexual reproduction, death. The unique inversion occurs from any stage ephira, juvenile medusa, or adult medusa, which can return directly to the polyp stage. Thus short-circuiting programmed death and obligatory sexual reproduction. We start from the medusa to the cyst. This is dedifferentiation. Dedifferentiation is triggered during stress, such as hunger, injury, or temperature changes, during cessation of locomotion and loss of swimming ability, during progressive retraction of the umbrella which constitutes a bell-shaped body. Morphological transformation operates through rheso-RBT-ion of tentacles and specialized structures. Then contraction of the body restructuring of the cyst occurs via complete reorganization of cellular architecture. Then, formation of new developmental axes and reactivation of embryonic signaling pathways. WNT. Hedgehog. Notch genes. The polyp must emerge, which includes development of a stolon, therefore a stem for attachment to the substrate. Next comes formation of tentacles around the mouth. For this, reconstitution of the gastrovascular digestive system occurs. Finally, return to a functional juvenile state. The complete phase lasts to 2-4 weeks, depending on conditions. The critical phase, called cyst, lasts 7 to 10 days, and polyp emergence takes a few days. In most organisms, a specialized cell, for example, a muscle cell or neuron, can only transform into another type of specialized cell by passing through a stem cell state. However, Turritopsis Dohrnii is capable of a shortcut. Transdifferentiation allows an already differentiated cell to transform directly into another type of differentiated cell, without having to return to a pluripotent stem cell state. It is this mechanism that allows the medusa to transform the cells of its bell and tentacles directly into polyp cells, thus erasing its age and restarting its life cycle. This phenomenon is extraordinarily rare among metazoans, which are defined as multicellular organisms. And this is what makes Turritopsis Dohrnii a unique and essential subject of study for research on aging and regeneration. The comparison between Turritopsis Dohrnii and Turritopsis rubra is indeed at the heart of recent genomic studies. Studies have shown that Turritopsis Dohrnii maintains a very high rejuvenation potential, up to 100%, even after reaching sexual maturity and reproducing. It is this efficiency that earns it its status as a biological immortal. Observations and studies on Turritopsis rubra are more contrasted. Initially, certain research assumed it was incapable of post-reproduction rejuvenation, but more recent studies have highlighted that it can actually regenerate, but at a rate significantly lower than that of Turritopsis Dohrnii. Its reverse capacity is therefore less efficient. In 2022, a major study published in the journal PNAS compared the genome of these two species to understand what makes Turritopsis Dohrnii so unique. Genomic analysis revealed that Turritopsis Dohrnii possesses twice as many copies of genes linked to DNA repair and protection than Turritopsis rubra. These genes play a crucial role in producing proteins that protect cells from aging and damage. Turritopsis Dohrnii also showed amplification of genes involved in telomere maintenance, the protective caps of chromosomes. Telomere shortening is a major cause of cellular aging, and Turritopsis Dohrnii's ability to maintain them is a key element of its perpetual youth. Scientists chose Turritopsis rubra as the ideal converter because it is a close genetic relative of Turritopsis Dohrnii, but without possessing the same rejuvenation efficiency. This comparison allows isolation of genes and mechanisms that are specific to Turritopsis Dohrnii's biological immortality. Genes linked to telomere maintenance are more numerous in Turritopsis Dohrnii. Also, more cellular stress signaling pathways are amplified, and cell cycle control mechanisms are modified. Turritopsis Dohrnii's selective advantages are survival capacity in unpredictable environments, multiplication of reproductive opportunities, and exceptional developmental plasticity. The comparison between these two species is not intended to say that one is mortal and the other immortal, but rather to understand why one excels in rejuvenation capacity, a trait that its cousin possesses in a much less pronounced manner. Among DNA repair, we have documented overexpression admitting the PARP family poly ADP ribose polymerase for single-strand break repair. double-strand breaks. Then, the nerve system nucleotide excision repair eliminates DNA adaptase. The functional impact allows maintenance of genomic integrity during repeated transdifferentiation cycles, avoiding accumulation of deleterious mutations. Next, telomere maintenance and its amplified genes. TERT is telomerase reverse transcript SE, a catalytic enzyme. TERCT is telomerase RNA component, shelter in proteins for protecting chromosomal ends and telomerase regulators for controlling enzymatic activity. All this favors prevention and hyperactive W and T signaling of telomerase shortening, avoiding classic replica T senescence. Hyperactive W and T signaling includes overexpressed components, including catenin, central effector of the pathway, WNT3, WNT8, more O general ligands, TCF-left, target transcription factors, and GSK3 inhibitors for catenin stabilization. It has multiple roles, including cellular reprogramming during inversion, maintenance of stem cell PL pluripotency, and control of the polyp medusa developmental axis. Then, stem cell renewal intervenes in PL pluripotency factors, including SOX2, October for homologs for maintaining undifferentiated state, KLF4, CMYC for cellular reprogramming, and non-OG for resistance to differentiation. Cell cycle regulators occur. Cyclin CDK for controlled progression of division, and modified P53 for adjusted survival apoptosis balance. Regarding the reinforced antioxidant system, overexpressed enzymes are therefore superoxide dismutase called SOD intervening in neutralization of superoxide radicals. Catalon set for hydrogen peroxide decomposition. Blood ionic peroxidase generating protection against lipid stress. Thiodoxin for desulfide bridge reduction. And protective molecules including HSP called heat shock proteins comprising anti-aggregation chaperones. Blood ionic. Major cellular antioxidant. These gene amplifications create an integrated network allowing genomic preservation during transdifferentiation stresses. Unlimited regeneration without damage accumulation. Developmental flexibility for cycle inversion. And resistance to cellular aging via antioxidants. This unique genomic architecture makes Turritopsis Dohrnii an exceptional model for understanding molecular mechanisms of biological immortality. Turritopsis Dohrnii possesses a genomic architecture where coordinated amplification of DNA repair genes, telomerase regulation, and Yamanaka factors establishes a cellular environment constitutive life permissive to biological immortality. Longevity networks including SIRT3, anti-senescence pathways, and cellular plasticity are pre-activated rather than IUCI suggesting specific evolutionary selection for indefinite cellular maintenance. The multiplicity of genomic protection pathways create systemic molecular robustness where failure of one mechanism is automatically compensated by redundant systems. The permanent presence of reprogramming factors confers intrinsic developmental plasticity, transforming life cycle inversion into a direct, emergent property of the genomic system. Turritopsis Dohrnii genome is structurally organized around cellular maintenance rather than programmed senescence, making biological immortality a logical consequence of its molecular terrain rather than a punctual adaptation to environmental stress. Yamanaka factors October 4, SOX2, KLF4, CMYC allow artificial reprogramming of differentiated cells toward a PL pluripotent state in mammals, a process that Turritopsis Dohrnii naturally masters during its transdifferentiation. In Turritopsis Dohrnii, coordinated reactivation of embryonic genes and simultaneous reduction of aging markers, spontaneous Turritopsis is Dohrnii manifests. TS extreme cellular plasticity, where highly differentiated cells can be reprogrammed toward totally distinct lineages, even exceeding the capacities of human-induced pluripotent stem cells named iPSCs. Strict control of this plasticity constitutes the major challenge for therapeutic transposition, as uncontrolled reprogramming can lead to tumor formations, as observed in certain experimental protocols of cellular rejuvenation in mammals. Indeed, this natural mastery of the plasticity stability balance makes Turritopsis Dohrnii a crucial study model for developing secure cellular reprogramming strategies for regenerative medicine. In Turritopsis Dohrnii, the capacity for ontogenic reversion, that is, the return from a medusoidal stage to a functional polyp stage, does not confer physiological invulnerability. The organism remains subject to external lethal factors such as interspecies predation, fish, cnidaria and S, crustaceans, pathogens, bacteria, protozoa, viruses, parasitic infections, as well as deleterious effects of chemical and particulate pollution, heavy metals, microplastics. These biotic and abiotic constraints frequently interrupt the life cycle or complete implementation of the reversion process. Environmental parameters strongly influence the probability of cycle recurrence. Variations in temperature, salinity, dissolved oxygen concentration, or trophic availability can alter tissue viability and inhibit the cellular plasticity necessary for transdifferentiation under controlled experimental conditions. Notably in Shin Kubota's work, certain specimens have presented up to 10 successive reversion cycles, demonstrating that the limitation is not of intrinsic physiological order but environmental. However, in situ observations indicate that a very reduced proportion of individuals reaches a second complete cycle under natural conditions. Thus, Turritopsis Dohrnii immortality must be understood as a conditional biological potentiality, rarely actualized in the marine ecosystem due to the convergence of ecological constraints and selective pressures. The jellyfish Turritopsis Dohrnii constitutes a biological model of choice in the fields of gerontology and regenerative medicine due to its unique capacity to invert its life cycle. This phenomenon, qualified as transdifferentiation, allows the organism to regress from a sexually mature adult state toward a juvenile polyp state, thus escaping senescence and death. Elucidation of underlying molecular mechanisms offers an exceptional investigative framework for anti-aging therapies and tissue regeneration. Scientific interest in this jellyfish resides in analysis of cellular reprogramming processes that convert its functional immortality. Current research concentrates on several axes. On one hand, genomic integrity and telomerase. Genomic studies have revealed overrepresentation of genes involved in gene and age repair and regulation of the telomerase enzyme, whose activity is crucial for maintaining telomere length. Markers of senescence. On the other hand, cellular plasticity and reprogramming. The transdifferentiation process involves dedifferentiation of somatic cells into more primitive cells, a phenomenon homologous to reprogramming induced by Yamanaka factors in mammals, and which relies on selective ray activation of embryonic development genes and signaling pathways specific to pluripotency. Despite its potential, transposition of Turritopsis Dohrnii discoveries to human biology encounters major obstacles. Human genomic and tissue complexity is superior, and aging is a systemic process, which is not comparable to rejuvenation of a simpler organism. On the other hand, ray does not constitute a model for direct gene transplantation, but rather a source of inspiration and a biological proof of concept. Study of its mechanisms opens new research pathways, such as development of secure reprogramming therapies and exploration of longevity genes, homologous to those of the jellyfish. In conclusion, if the immortal jellyfish does not provide a direct solution, it offers a paradigm for studying aging inversion, thus pushing back the frontiers of biology and regenerative medicine. Turritopsis Dohrnii, I represent the unique organism capable of fundamentally reprogramming its cellular destiny, inverting the aging process through natural mastery of transdifferentiation that human science is still trying to decipher. Studying this immortal jellyfish allows us to map the biological frontiers of the possible and identify molecular mechanisms that govern cellular rejuvenation. Each discovery about its unique genomic architecture illuminates potential pathways toward controlled manipulation of aging. Understanding Turritopsis is Dohrnii's genetic networks, Yamanaka factors, Telomerase regulation, extreme cellular plasticity opens revolutionary therapeutic perspectives for slowing, even partially inverting our own aging. What if our deep understanding of this jellyfish could help us develop secure cellular reprogramming strategies, transforming the fiction of biological immortality into accessible medical reality?